Welcome to this explainer. Today, we're doing a deep dive, a real cinematic exploration into the exact molecular mechanisms that control your rest, your stress, and honestly, your entire nervous system. We're going to strip away all that superficial, generic sleep advice you've heard a million times and expose the raw cellular and neurological reality of how your body actually recovers. So think about this. Is sleep just a passive shutdown state? Like, are you just unplugging a computer for the night? Actually, no way. In reality, it's this highly active, explosive period of neurobiological and metabolic cleansing. It is literally a digestive event. Your brain fires up its glymphatic system to actively wash away the metabolic waste and the emotional residue we just naturally accumulate all day long. Now, to figure out why this vital cleansing process sometimes totally breaks down, we have to talk about allostasis. Basically, this is your body's way of finding physiological stability through change. When a stressor hits, your hypothalamic pituitary adrenal axis, the famous HPA axis, orchestrates this massive symphony of hormones, like cortisol and adrenaline, just to keep you alive. It's a brilliant evolutionary adaptation, right? But there's a fatal catch. See, we're biologically hardwired to oscillate seamlessly back and forth between two main states. On one side, you've got the sympathetic fight or flight mode, which is driven by high cortisol and low heart rate variability. On the flip side is the parasympathetic mode, driven by vagal tone, where high HRV and deep restorative cellular repair happen. But here is the modern tragedy. We humans get caught in an autonomic stress trap. We are literally locked in chronic physiological overdrive. And being stuck there creates a catastrophic biological sequence. First, chronic daily stress triggers systemic inflammation. Then, your HP axis, which is just facing this relentless, continuous activation, starts to burn out. Cortisol levels, which are supposed to plummet at night, become completely unmoored and erratic. And the end result? Your restorative sleep architecture is completely destroyed. You might be unconscious, sure, but your cells, they are not resting. When you're trapped in that sympathetic overdrive, you block something absolutely vital. Remember how we said sleep is a digestive event? Well, during deep parasympathetic rest, this incredible thing called the migrating motor complex activates. It's like a rhythmic wave in your gut that clears out metabolic waste and balances your microbiome. But if your nervous system is even slightly stimulated, that biological street sweeper never even turns on. So how do we actually fix the hardware? Well, instead of just looking at herbs as generic supplements, we need to categorize them by their specific neurological targets. Adaptogens target the macro-HPA axis. Nervine relaxants go straight for the central nervous system for rapid, immediate calming. And nervine tonics, they physically target and repair exhausted nerve tissues over time. Let's dive into adaptogens first. The absolute defining characteristic of an adaptogen is that it's a bidirectional master regulator. It's not a simple stimulant. Think about coffee. Coffee just forcefully pushes your body up, right? But an adaptogen, like ashwagandha or rhodiola, actually reads the biological room. If your cortisol is spiking, it buffers it. If your stress response is completely tapped out, it restores it. It normalizes those erratic circadian rhythms to help build long-term metabolic resilience. And what's truly fascinating is watching how this plays out at the molecular level. Take rhodiola, for example. It doesn't just, calm you down. Clinical data actually shows it reduces the expression of the C-Phos gene right in the hypothalamus. By downregulating this specific neuronal marker, rhodiola literally alters your genetic expression to stop the stress cascade right at its source. That is a real game changer. Okay, so while adaptogens handle that macro-level hormone cascade, nervine relaxants operate at the micro-level, specifically dealing with GABA. Gamma-aminobutyric acid is the primary inhibitory neurotransmitter in your brain. It is your neurobiological brake pedal. When GABA binds to its receptors, a flood of negatively charged chloride ions rushes into the neuron, inducing hyperpolarization and suppressing that overactive firing, so you can actually drift off to sleep. This is exactly where specific nervine herbs interact directly with your neurochemistry. It's incredibly precise. Valerian actually docks right onto the beta subunit of the GABA-A receptor, which inhibits the breakdown of GABA. Passionflower and chamomile use this powerful flavonoid called apigenin to dock at the exact same receptor sites that pharmaceutical sedatives use. Meanwhile, lemon balm smoothly shifts the autonomic system into a parasympathetic state, easing that acute nervous tension. Now, you might be wondering, if they hit the exact same receptors as synthetic drugs, why aren't they dangerous? Well, it all comes down to binding affinity. Synthetic sleep drugs are what we call full agonists. They violently slam the GABA receptor wide open, which is what causes that severe morning grogginess and chemical dependence. But these plant flavonoids, they are partial agonists. They gently stimulate the receptor, just enough to induce anxiolysis and sleep, but without the severe cognitive impairment.
But what happens if your nervous system isn't just overstimulated, but actually structurally exhausted? That's where Nervine tonics come in. We call these trophy restoratives. Herbs like milky oats and American skullcap don't just temporarily sedate you. Instead, they slowly, methodically nourish and rebuilds the frayed physical insulation of your depleted nerve tissues after a period of chronic burnout.
So, does all this ancient plant biology actually translate into modern clinical data? Absolutely. Take a look at this number, -0.84. This is the standardized mean difference, pulled from a really rigorous meta-analysis looking at single herb interventions. And let me tell you, in the realm of clinical statistics, -0.84 is a massive, highly significant therapeutic signal. It proves, objectively, that these compounds profoundly improve sleep metrics. And here is something super surprising. Single herb interventions actually outperformed those combination herbal remedies you see everywhere. Chamomile and lemon balm consistently demonstrated the absolute strongest therapeutic signals for resolving insomnia. It turns out that a targeted, high-quality dose of a single botanical compound interacting perfectly with your GABA receptors is often way more effective than a scattered shotgun approach. However, because this explainer is dedicated to first principles science, we really have to look at the other side of the coin here. Because these plant compounds can alter deep genetic expression and endocrine function, more is not always better. If you rely too heavily on high-dose botanical interventions without actually fixing your underlying lifestyle stressors, it can lead to some very real, very dangerous neuroendocrine consequences. Let's walk through a pretty sobering clinical case study so you can see exactly what I mean. A 55-year-old woman was consuming 950 milligrams of ashwagandha extract every single day. Fast forward 1 year, and she developed progressive weight gain and Cushingoid features. The diagnosis? Her basal cortisol had plummeted below 0.5 micrograms per deciliter. She had actually induced secondary adrenal insufficiency. Even 6 months after she stopped taking the herb, her HPA axis was still suppressed, and she needed continuous pharmaceutical hydrocortisone just to function. Listen, these are incredibly powerful biological tools, and they absolutely command respect. Furthermore, maintaining scientific integrity means we also need to acknowledge the limitations in the current clinical trials. While the biological mechanism of how these plants interact with the HPA axis is undeniable, a lot of the trials on herbal sedatives still suffer from small sample sizes and a high risk of bias. The science here is immensely promising for sure, but we are still learning how to accurately measure the full impact of these complex plant matrices. So what's the big takeaway from all this? Let's synthesize everything into one unified biological framework. Step 1. Use targeted adaptogens to modulate the macro HPA axis and buffer your daily cortisol curve. Step 2. Bring in GABAergic nervine relaxants in the evening to ease that acute synaptic tension. Step 3. By doing this, you allow your autonomic nervous system to smoothly shift into a parasympathetic state, which activates that vital migrating motor complex. And finally, step 4. This entire physiological cascade unlocks true, deep, restorative neurobiological sleep. I want to leave you with one final, pretty profound question to think about. As we aggressively try to optimize our neurobiology to handle modern chronic stress, we really have to ask ourselves, are we genuinely adapting to our modern environment, or are we simply utilizing ancient plant biology just to survive it? True optimization is about aligning our modern lives with the evolutionary rhythms our bodies were actually built for. Thank you so much for joining me on this explainer. Keep questioning, keep learning, and I'll see you next time.